Hello everyone, my name is Felipe and I'll be presenting Bridging Biology and Behavior Epigenetic Pathways of Change. So make sure to take notes because there will be a quiz after the presentation. No, I'm just kidding. I'm a recording, I can't give you a quiz, so you guys are safe. You're lucky I wasn't able to show up today. Now, first things first, I want to introduce epigenetics to you guys. The definition of epigenetics is the change in an organism caused by modification of gene expression rather than an alteration to its genetic code. Now, for us to really understand that, let's put it into some context so we can understand it a little bit better. I want you to think about that nature versus nurture debate. So we can think of two siblings. These siblings are identical twins. Their genetic code is the exact same when they're born, but as life goes on, maybe one twin starts to get really into running marathons. The other one, not so much. He lives a more sedentary lifestyle, which is fine, it's fine, sedentary life. Another difference, one twin eats really healthy foods, and the other twin he just can't fight that patty melt Whataburger craving. But who can, right? So over time, these things are going to lead to drastic differences in how they look. Of course, right? These are really big differences. After all, they're going to make a big change. But what epigenetics tells us is that these habits not only affect looks, but also their genome. Now, to find out just how epigenetic changes can affect our genome, we first have to do a little review of DNA transcription and translation. So we're going to flash back to 9th grade when we learned about biology and DNA, and we're going to remember that. Our cells have DNA, right? Which is the genetic code that is necessary to make proteins that make us. So remember, inside DNA are genes, which are specific segments that code for a specific protein. Now, specifically during DNA transcription, our cells copy this DNA strand as RNA, and then during translation, this RNA copy is read and translated by a ribosome to create that protein. So all cells have the ability to code for all the proteins that make us up. But during transcription, these epigenetic factors help cells differentiate between the proteins that they need to make and the proteins they do not need to make. So this would be the translation portion of it. Now that we have some context on what epigenetics is, we can dive a bit further. I mentioned cells can differentiate which genes not to transcribe with epigenetic markers. But what are those epigenetic markers or epigenetic factors? Well, the answer is methylation and acetylation. First, let's break down methylation. In this process, a methyl group, methylation, is added to a section of DNA, a gene. This addition of the methyl group blocks the transcription of that gene. Don't worry, demethylation is another process in which a methyl group is removed, and this allows transcription, which in turn means that the gene is expressed. On the other hand, acetylation does not affect DNA directly. What it does is it affects these things called histones, which are little proteins that DNA wraps itself around. Now, acetylation, when it's added, it allows histones to be more loosely packed next to each other. Now, this space, this extra space, allows DNA to be transcribed more easily. Versus if there is not an acetyl group attached to the histones, they'll be more tightly packed, and this won't allow DNA to be transcribed as easily, which means there won't be transcription of that gene, and we won't have that protein made. So, in summary, methylation turns gene expressions off, and acetylation turns genes on. Now, good with that. And at this point, I'm going to imagine someone's in there asking, now Julio, why would we need or want to turn off genes? Don't we need to make proteins for metabolism and whatnot? And I want to thank whoever asked that because it's exactly what I'm going to talk to you guys about. So imagine how a cell in your stomach can produce hydrochloric acid for digestion, right? Well, so can all the cells in your body, theoretically. Even cells in your eyes have the genetic code for producing hydrochloric acid. But the cells in your eyes are smart enough to not transcribe that gene so that your eyes don't produce hydrochloric acid. It's a good thing, right? We don't want hydrochloric acid in our eyes. So, again, all cells in your body theoretically have the ability to produce acid. It's part of your genome, right, of your DNA, but epigenetic markers signal cells that are not supposed to be producing things like hydrochloric acid to not transcribe that gene. This is how genetic expression is modified without altering the genetic code. So, some things can affect our epigenome, the combination of epigenetic markers and our genome. Some things that can affect that are diet, chemical exposure, medication, diseases. Basically, our whole environment directly affects our gene expression. We have found some correlation between diet and genetic expression and victims of famine, for example, in which people that were exposed to famine during an early gestation exhibited hypomethylation of the IGF-2 or insulin-like growth factor 2 gene that is important for making a protein that is really, really important for fetal growth, development, and cell proliferation. Now, all these things, right? Fetal growth, development, cell proliferation, they're really, really good. But if there is an excess of IGF-2 activity, this is associated with various cancers like liver cancer, breast cancer, lung cancer. Another factor, smoking. Smoking is another habit that can impact DNA methylation. So in smokers, we have found that the AHRR or aryl hydrocarbon receptor repressor gene is hypomethylated, which means cells do not have the protein that controls cell growth and division, which then causes uncontrolled cell growth, or AKA cancer. Now, I don't want you guys to worry if you do smoke or if maybe your eating habits aren't the best. I have good news for you. The good thing about our bodies is that they can reverse methylation. Remember, there's demethylation and deacetylation. This means that by improving our habits, we can improve our epigenome. For example, that study that I mentioned about smoking and methylation of the AHRR gene also found that the levels of methylation of the AHRR gene returned to normal levels after some years of smoking cessation, although this was dependent on the amount of smoking and the length that the person smoked for. The same thing for our diets. If we try to fix our diet, then our epigenomes can also be repaired. So like I said, I don't want to leave you guys with any worry or nervousness about your genetic code now, but what I do want to leave you guys with is this. Although our body is affected by our environment, it's ultimately up to us to make changes in our lives if we want to see improvement. These things are not permanent. Now, these things can be applied to genetics, any other part of life. Your entire potential is literally encoded within you, within all of us. But the choices and the actions that you take are the key to unlocking your fullest potential. Now, Here are my works cited. I'll make sure that the links are provided in the description, but I can't promise access because some of them were obtained through the HCC library database. Shout out HCC. If anything, I'll try to find free access links for you guys, but that is all I have for you today. Are there any questions for me? I'm just kidding, guys. Again, I'm a recording. You can't ask me questions, but I wish you could. Thank you guys so much for listening. Appreciate it.